systematic reviews and meta-analyses, and we're going to do it through using this particular paper. I'm going to run through 4 or 5 slides that talk about the process of evaluating the validity of a systematic review, and then we will systematically look at this paper. Okay, it'll take us probably less than 30 minutes to get all that done. Hopefully have you out of here by 1 o'clock. This is your last thing today, right? Hopefully we'll have you out of here by 1. So we've been talking in this evidence-based medicine concentration, we've been talking about how do we determine what the strongest evidence is for anything in medicine. We've covered diagnostic testing, we've covered therapies, we're going to cover prognosis and harm in the future. But systematic reviews and meta-analyses are the strongest evidence that we have available to us on any particular subject in medicine. Okay? And so, one of the reasons that systematic review and meta-analyses are the strongest is that they are a systematic review or a meta-analysis of any particular area in medicine, whether it's a therapy, a diagnostic test, an issue of prognosis or harm. They contain rigor in gathering all the pertinent information, which are the studies, on a particular issue. And they use the same rigorous analyses of papers that we looked at when we talked about therapy and diagnostic testing. So what happens in a systematic review is 2 or 3 of us get together and say, I wonder what the evidence is on trying to get the blood pressure down in patients. You know, we know that nearly 40% of people in the world who have diagnosed high blood pressure do not have their blood pressure at normal levels. 40%. And we know that it causes heart attacks, strokes, early death. I mean, this is a big time issue. So let's try to figure out what's the evidence out there for how we can intervene on our patients to try to figure out how to get the pressures down. And so we spend a lot of time up front if we're going to be doing this research, brainstorming what it is we want to look at. And probably we're researchers in the area of hypertension, so we know this literature really well. And we come up with some things that say, well, we only want to look at randomized controlled trials because that's the strongest evidence there is on individual studies. We only want to look at randomized controlled trials. We want to look at every randomized controlled trial there is out there. Doesn't matter what language it's in, it doesn't matter which country it comes in. We want to look at everyone out there. And we want to subject every one of those randomized controlled trials to the same rigor we would apply to any individual trial. We would ask those same questions about validity that we would ask of any of those individual trials. And we'd say, well, where are we gonna look? Well, we'll decide we're going to look at Embase, we're going to look at Medline, we're going to look at all these other places, and we're going to try to find every single trial out there. Can we do that? Maybe not, but we can get really close. Okay? The advantage over individual studies are that they tend to further minimize bias because you're comparing, you're aggregating the results of many more numbers and you're subjecting them to an analysis. You're selecting only randomized controlled trials, so you minimize bias, seeking all the published and unpublished studies, and you reduce random error by increasing the numbers. So if you look at individual trials on hypertension, you might have a trial that looked at 1,000 patients. You know, you might have a trial that looked at 900 patients. The paper you're looking at right now, by combining all of these, by doing this very systematic approach, had 55,000, almost 56,000 patients in them. So by doing that, you reduce the random error. We're just describing how these are done and why they're more important than individual clinical trials. Okay, so these are the questions that we have to run through when we're trying to figure out, is this systematic review a valid systematic review? Number one, is this systematic review or meta-analysis of randomized controlled trials? Two, does it contain a comprehensive and detailed search of the relevant trials? Does it tell you exactly what they looked through to get these trials done? Do the individual studies assessed for validity? We seek out evidence that they went beyond looking for standard bibliographic references. Did they do a hand search of journals, conference proceedings, and theses? Otherwise, one might miss 50% of the data that's out there. The methods section should also contain a statement about how they assess the validity of individual trials. And then you want to know, is this evidence important? Are the results consistent across studies? What's the magnitude of the treatment effect? How precise is the treatment effect? These are the same questions we ask on every individual trial, right? When we ask what's the magnitude of the treatment effect, we were looking for a mean difference between the control group and the experimental group, or we were looking for an odds ratio, or we were looking for some sort of a difference, figure out what the magnitude of the treatment effect was. How precise was the treatment effect? Can anyone remember what we look at for precision of the treatment effect? 95% confidence interval. You get a free lunch today. McNeil's going to pay for it. Okay, so that's the preciseness of the treatment effect. And we're going to introduce a new thing called a forest plot today, because the forest plot is a graphical presentation of all of these studies and all of this data. And if we learn to read a forest plot, we can make some analyses, some decisions about whether or not this is important information pretty quickly, and we'll go through that, okay? This is a forest plot, but we'll go through that in a few minutes. At the end of your paper, there should be one of these forms. It's the same form that you've been using for evidence-based medicine. And what we're going to try to do is go through and fill this out based on what you've just read on the methods. And the first question is, were the question and methods clearly stated? And were the search methods used to locate relevant studies comprehensive? Okay? And you've read these methods. So what is the question here? You know what the question is? Yeah, the effectiveness of ways of intervening at the patient level, at the provider level, and at the systems level. What's the effectiveness of intervening to further get those blood pressures down to in the normal range? That's really the question. And they've divided these studies up into things that intervened on the patients, things that intervened on the provider, and things that intervened on the system. Okay, so that question is pretty clearly laid out early in the paper. Did they talk about where they went and how they searched for the universe of studies that speak to this? They did. They said they went to Medline, and they said they went to Embase. What is Medline? Does anyone know? It's a database. It's a huge database, maybe the largest database in the world. It sits and is managed by the National Library of Medicine in Bethesda, Maryland. So it's a National Library of Medicine, federal government funded database that contains many of, or most of, the biomedical, human-related research studies done in the world. So it's a huge database. Embase is privately owned. It's mostly concentrated on drugs. It's published and modified by Elsevier, which is a publishing house. 32 million records Embase has. 32 million records covering 8,500 journals. So it's also a very large database. Now they did two other things. Do you remember the two other things they did? They did clinicaltrials.gov. So if you just type in clinicaltrials.gov, it'll take you right to a website that's available to everybody, and you can type in blood pressure, you can type in glioblastoma, you can type in lung cancer, and it will list every trial that's ongoing or has just finished or is getting ready to start that's been registered with the National Institutes of Health. So it's a good place to go. What they did that for was to try to figure out what studies are currently or recently stopped that haven't yet appeared in the journals. So they're being a little bit more comprehensive. They did one other thing.
Studied unpublished trials?
They looked for unpublished trials. That's a much harder thing to get at, but they looked for unpublished trials. The reason that unpublished trials are important is that negative outcomes are, there's a publishing bias, there's a publication bias toward negative outcomes. So if you decide to do a study and you do a randomized controlled trial and it turns out that whatever you tried to do didn't affect anything, there was no significant difference between your control group and your therapeutic group, it's not as likely to get published because there's a publication bias toward positive outcomes. So that's the reason to look for unpublished data. And then they're looking for, they did a hand search on the bibliography of every paper they found. Now you can imagine that process alone takes hundreds, if not thousands of hours. I did a meta-analysis with a fourth-year medical student one time on one very narrow subject, and it took us 6 months to complete finding all the trials before we even started analyzing them. I mean, we weren't doing it full-time, it wasn't our full-time job, but we were both working on it quite a bit, so it takes a lot of hours to get this done. So we think they were pretty rigorous at trying to find all the studies, right? And they told us what they were doing. Number two, were explicit methods used to determine which articles to include in the review, and were the selection and assessment of the methodological quality of the primary studies reproducible and free from bias. That's a lot of stuff. That's a lot of stuff in there, but let's take it one piece at a time. What explicit methods were used to determine which articles to put in? Now, if you look at page 111, you've got a flow chart. And this flow chart tells you how many they started with and why they were excluded. This is a really nice chart to review to help you decide how they went about getting down to the 100 articles that they included in their thing. And you can ask yourself the question, if you're doing this review, you can quickly look at this chart and say to yourself, are those reasonable reasons to exclude them? Citations excluded by title and abstract review, 5,000. Articles excluded, 858. Some because they're not a randomized controlled trial, some they're not adult hypersensitive patients. They're only looking at randomized controlled trials. They're only looking at adult patients. They're not looking at pediatric patients. So if they didn't meet the criteria that they had already set out, they're excluded. Okay? So trying to answer the first part of that study, how many investigators are doing this study? Two people are trying to do the whole thing. Each of the investigators reviewed every article, and if there was agreement, it was just done. If there was disagreement, how did they decide whether to keep it in or throw it out? Probably the strongest personality won. Or the preferred professor won and the student lost. Or more commonly, they have a dialectic, and they discuss this, and they argue back and forth, and then they decide on a consensus. Okay. It also asks about the assessment of methodology quality. And there's a statement on page 112. Probably ran right by it because it means nothing to you. It says, let's see. It's not. No, it's on page one. Yeah, it's on page 112, and it's about midway down, right about here on the page. And it's a paragraph that starts out, to assess quality, we modified the Cochrane risk bias tool to make it applicable to cluster trials in implementation research. That doesn't mean anything to us, except for the fact that they actually applied some tool that we don't need to understand very well. But they did, in fact, apply a tool that has been validated by someone, and we might know about it if we were researchers. They applied a tool to minimize bias, and that increases the quality. So it speaks to this question: did they tell us what their methodological quality was? Were the primary studies reproducible and free from bias? Well, we just talked about quality and bias. They used that Cochrane thing. How about reproducibility? Reproducibility we would know that by saying, well, did the studies look, did the outcomes look fairly similar? And there's a table that usually tells us that. Look at table 2 on page 114. On the left side, so this is where you've got the implementation strategies, you've got the patient level, the provider level, and the multi-level. You've got all three strategies that they group these papers into. The number of studies listed under each one, how many participants there were under each one, and then you've got range of ages and ranges of percentages of men and women, and then you've got range of baseline blood pressure. So if you just take your eyes and run down those rows or columns of numbers, you're asking yourself, are these fairly similar, or are they so different that they're comparing apples and oranges? And as I did that yesterday and earlier today, as I did that, I don't find anything that's so far outside the realm that I would say this is invalid. They seem to be fairly similar groups of people. Their starting blood pressures are similar. There's a little bit of difference in percentages of men, but that's to be expected if we're going to include VA data. So yeah, they're pretty reasonable, comparable studies. Were the differences in individual study results adequate, and were the results of the primary studies combined appropriately? There again, table 2 tells us that they are comparable groups of people. They've combined them appropriately, so we can proceed. The results were a decrease in systolic and diastolic blood pressure. So here's where we look at the forest plot. look at page 116, and you'll see this forest plot. If you go to the bottom of this forest plot, you see there's a 0 to the left is a minus 15. That's a minus 15 millimeters of mercury. To the right is plus 15 millimeters of mercury. And every study, or every group of studies, is set there in those squares. So you can look at this forest plot and say, how many of these studies had an increase in blood pressure? How many of these squares are on the right side of that vertical line? None.
None of them.
How many are clearly on the right side or on the left side of that vertical line without touching it? Almost all. There are 2 that are touching it. So by glancing at this forest plot, you can tell that every group of studies they looked at, when they combined mean reduction of blood pressure, which is what those squares are, the outcomes are mean reduction of systolic and mean reduction of diastolic. Every one of those falls to the left side of the line. So this is meaningful data. Now, whether or not those distances from that vertical line are statistically significant, we can't tell, because they don't give us that on the forest plot. But in the paper, they tell us that they're all statistically significant, with the exception of electronic reminders for the provider. Well, when they intervened on providers, none of them were statistically significant. When they intervened on the system, they were all significant. When they intervened on the patients, they were all significant. It was just the providers that weren't so trainable. What does that mean? Maybe it means the providers weren't trainable. Maybe it means the providers were already doing a damn good job. I don't know. I choose to say the latter, but it doesn't matter because it's not an outcome of this study. But if we're going to implement something like this in our practice, we know there's good data to intervene on the patients and on the system. Okay, so figure 3, I think is on page 117. This looks like a table. It's actually a figure, but it's table 3. And at the top, you've got team-based care with titration by non-physician. So what this means is you've got a provider, you've got nurses, you've got other people in the office, and you've got a team-based approach. And maybe there's titration by a non-physician. Titration of medication type and titration of medication dose by the non-physician was the most powerful drop in systolic blood pressure. These are listed according to effect size. And up there in the upper left, it says team-based care with titration by non-physician. So when the PA was titrating the dose, or the nurse practitioner, or the nurse in the office, whoever the non-physician was, they did a better job for some reason at getting the pressure down. And then team-based care with the physician titrating it, and then multi-level strategy without team-based care. So this is a nice way of listing effect size. Look at page page 113. On the right side of the page, it says team-based care. Physicians and non-physician providers titrating medications had the largest reductions in pooled mean systolic blood pressure. 5.7 millimeters of mercury with a confidence interval of 3.6 to 7.9. What is the 5.7 in that paragraph? What is the 5.7 millimeters of mercury? It's the reduction in systolic blood pressure in all of the studies combined, in all of these 55,000 patients, when there was team-based care with titration by the physician or non-physician. Okay? We were able to bring the systolic pressure down almost 6 millimeters of mercury. With a 95% confidence interval of 3.6 to 7.9. What does that mean again? Someone want to articulate what that 95% confidence interval means?
5% chance it was 5% chance it was due to chance.
That's the p-value.
Oh.
95% confidence interval means that if you were to repeat this study, this systematic review, 100 times, 95% of the time you would find a mean reduction of systolic blood pressure of 3.6 at the lowest end or 7.9 at the highest end. So is 3.6 reasonable? It's an effect. Is 7.9 reasonable? It's a bigger effect. What we found was 5.7. So if it gives us confidence that this data is meaningful, okay? And it's, in my opinion, it's probably the most powerful information when you're reading a research study, the 95% confidence interval, because it's at least 3.7, it's at maximum 7.9. The mean average was 5.7. And the p-value was less than 0.001. What does that mean?
That was due to chance.
Yeah, it's less than 1 in 1,000 chances that this is due to chance alone. The probability is less than 1 in 1,000 that this is due to chance alone. So it's statistically significant, and we can probably rely on it. Okay. You will have a chance to review a meta-analysis or a systematic review. How are meta-analysis and systematic reviews different? They're not much different. You answer the same questions when you're analyzing them. Meta-analysis almost always has, I think to be a meta-analysis, it has to have a process in the analysis of all the studies where they combine the results of the individual papers with a new statistical run. They apply a new statistical run to all this data. And there are specific ways that that's done that we don't need to worry about. But it tells us in the paper that they've done it. And the reason that's more powerful is that we now have data combined, statistically combined data on 55,000 instead of 10,000. Okay? All right, so let me back up another, oh my God, I got 5 minutes. Let me back up a little bit and reiterate what ought to be going on in your heads a little bit. We want you to know, to have gone through and experienced how to analyze some of these papers rigorously, which is why we're having these small groups. We give you a paper and you answer those questions and then get together and discuss them in small groups. That process is merely so that you know what the process is. We don't really expect you to end up in December able to do this, ever. And we don't expect you, when you're out in practice, to do this. Because you won't have time. Okay? I do it, I don't know, 4 times a year. I do it 4 times a year. I take 5 or 6 journals. I run through the front cover to see if there's anything I'm interested in. And when there's anything I'm interested in, I flip it over and I read the methods. And I know the answers, I know these questions, so I know how to answer them pretty quickly, and I know how to read those tables and stuff, to look at those pretty quick. In about 5 or 6 minutes, I can decide whether this is a validly done study or not, and then I can go figure out which one of these figures shows me the effect size. Is this important or not? And I can judge it pretty well, and I'm probably right, meaning I agree with all my peers, about 90% of the time. I miss it about 10% of the time. When I go to Journal Club at either Department of Internal Medicine or to the Residence Journal Club, I go in, you know, sort of dressed for battle, and I've got it wrong. Well, my wife tells me I get it wrong all the time, so it's not news. It's just humbling. But I can mostly get it, mostly get where I need to be. I don't do that very often. 4, 5, 6 times a year when I'm really interested in something, or when something comes up usually it's at a cocktail party, someone spouts off some thing and we get into a disagreement, then I gotta go find the paper and spend some real time to see who was right. But what if I'm in clinic? You need to know specifically how this is rigorously done, because there'll be some questions. There'll be some questions on pants about it. They won't ask you to do it on paper, but they'll ask you what a confidence interval is. They might ask you what a p-value is. They might show you a forest plot and ask you what it means. Those are the kinds of things we want you to be able to do. But if you're in practice, what are you going to do? This information that we're spending 12 months teaching you is all going to be wrong in 5 to 6 years. Most of it. All the therapy stuff is going to be wrong in 5 to 6 years. You know? The basic science is not going to change all that great, but this therapy stuff, man, there isn't one antibiotic used today that I used in residency. Well, Bactrim. That's not true. Bactrim. All the others are new. In the 1980s, I don't know how many new antibiotics. It must have been 60 new antibiotics came on the market. Stuff changes so fast. So what to do? I'm going to step out here and make a declaration. I didn't bring a copy, but I subscribe to a thing called Journal Watch. It's published by the New England Journal of Medicine. What the New England Journal of Medicine has done is gone out in academic medicine and hired a bunch of people to do this stuff. People who are really weird because they like doing this stuff, okay? They like spending lots of hours at night analyzing, was this valid, did they do it all right? And then they write summaries of it. And the summaries are published in Journal Watch. So I've got Journal Watch for General Internal Medicine. I've got Journal Watch for GI. I've got Journal Watch for Cardiology. And I get them every 2 weeks. And so I just buzz through there and I only circle the ones that I'm interested in. And then I read them. And I don't have to analyze the article myself because someone else did it. And I'm paying $60 a year to get them to send that to me. That's a pretty good strategy because at least I'm trying to keep up with what's going on. And it's pretty reliable stuff. If I had only one place to go as a practitioner, I'd go to Up-to-Date. You know, do any of you have a subscription to Up-to-Date? One? Okay, so let me tell you what Up-to-Date is. Up-to-Date was started by Duke and Harvard. And they put together a board, and then they went out and hired experts. And those experts hired a group of reviewers in the field. One of my buddies who's on the faculty at Duke, or just retired from the faculty at Duke, was hired by Up-to-Date to be in charge of infectious disease. And they paid him a big pot of money to recruit other people. And it has become so successful that it's probably the go-to thing out there in clinical medicine. What it does is not only tell you, if you go to UpToDate and go put in congestive heart failure, it'll tell you how it presents. It's like reading a textbook. How it presents, what the physical findings are, what the laboratory findings are, what the echo looks like, what the BNP looks like. And the data that they tell you about the BNP is the best evidence to date. And they update it every month. So every subject is updated every month. And it's done through the process we're using in EBM. It's just done by a whole bunch of different people. That UpToDate is paid. You can personally get a subscription to UpToDate for 1 year for $199. I can't imagine a better source of information when you hit the clinics. I bet it'd be the best $199 you ever spent. As you're going through your clinical years, you have a patient with X, Y, and Z. You come home that night and sit down at your computer and go to UpToDate, and you can read everything UpToDate has to say on that disorder in 20 minutes. And it is up to date. Okay, it's not like opening a textbook, which is already 10 years old. $199. $369, you can get it for 2 years. Not to be too precise or anything. We did look that up this morning, and Amber tells me it's $369. The biggest point I'm trying to make is that this stuff is so fleeting in terms of timeline that you need a strategy. If you're going to be a practitioner and you're going to do it right, you need a strategy, whether that's something that comes to you in the mail, whether it's something that you have as on a computer like UpToDate. Relying on your friends at work won't work. Relying on what you learned in PA school won't work. It'll go bad. Okay, so the next thing you're going to see is the faculty will hand you a systematic review or a meta-analysis. It'll have attached to it this form, and you will use the same method you used this morning. Read the methods section. Try real hard. You read the abstract first, so you figure out what's the question. And then read the methods carefully. That's what you'll need to read to be able to fill out that form. And we won't, when we get in small groups, we won't accept yes, no, maybe. We'll want to know where you found it in the methods section and what did you use to make it. Okay?